Historically, trying to treat a genetic disease has been a bit like trying to repair a delicate, intricate watch using a 10-pound sledgehammer. You know something is broken inside, but you don't have the tools to reach in and fix it safely. This timeline reveals a massive imbalance. Years are spent in slow lab discovery, leaving a fraction of time for patient application. Zooming into the cellular phase, the massive molecular tool fails to breach the selective barrier of human cells. Mapping a genetic mutation doesn't actually cure the patient if we lack the computational speed to design the medicine and the physical precision to get it where it needs to go. That speed barrier is what AI is currently dismantling. Neural networks now generate complex, three-dimensional protein structures in seconds, replacing manual pipetting with high-speed computation. OpenAI recently launched a frontier model built specifically for this. It's called GPT-Rosalind, named after the DNA pioneer Rosalind Franklin. Instead of mixing liquids in a petri dish and waiting to see what happens, this model reads the literal language of proteins and DNA. It reasons through biological interactions entirely within a digital framework, running thousands of virtual simulations to find exact molecular matches. By simulating years of traditional lab work in a matter of days, AI turns the grueling marathon of drug design into a high-speed sprint. But designing the perfect cure on a computer still leaves us with that second hurdle. How do we get that digital blueprint physically inside a patient's living cells? Researchers funded by the National Institutes of Health recently tackled this physical barrier by engineering what they call a mini CRISPR system. This structural comparison highlights exactly what they achieved. On the left is the standard bulky CRISPR molecular machinery. On the right is the newly engineered miniaturized version, a vastly smaller tool that actually boosts editing efficiency inside human cells. It is the biological equivalent of replacing a heavy-duty 18-wheeler with a nimble delivery drone that can easily navigate the microscopic tight spaces of the human body. By shrinking the physical size of the machinery, we can now deliver genetic therapies to tissues and organs that were entirely out of reach. Once that tiny drone is inside the cell, it faces another massive risk. If it cuts the wrong piece of DNA, the cure becomes an entirely new disease. To solve this, scientists published a study in Nature detailing a CRISPR variant called thermo-Cas9. Think of it as a highly specific, smart scalpel. Watch how thermo-Cas9 operates. It moves along the DNA strand until it spots a unique methylation pattern, acting as a red flag identifying the cell as cancerous. The tool ignores the uniform, healthy segments, locking on and precisely severing only the marked tumor DNA. Cancer treatment has long relied on systemic poisons that attack the whole body. This mechanism removes the tumor at the genetic level, leaving healthy cells completely untouched. We are also seeing these surgical tools target severe neurodegenerative conditions, specifically Huntington's disease. A recent study tested a set of self-switching CRISPR genetic scissors on mice. The goal was to target the root genetic mutation that causes Huntington's, rather than just managing the downstream effects. The researchers deployed these molecular scissors after the mice had already begun exhibiting physical symptoms of the disease. The intervention still successfully targeted the mutation. If this translates to human trials, patients would have a therapy that actively halts or reverses the deterioration of their brain by fixing the DNA itself, offering a real cure where medicine historically only offered comfort. Now, zoom out from a single disease to the entire medical pipeline. Everything we've covered, from the AI modeling to the physical CRISPR testing, is currently being linked together into a single automated loop. AI systems now execute thousands of lab experiments without human intervention, directly controlling robotic arms to manipulate vials and pipettes in sterile environments. These systems can run thousands of iterative tests a day, evaluating results and adjusting their next physical actions in real time, all without a human researcher ever stepping into the room. This closed-loop automation creates an astonishing volume of daily research, drastically accelerating the rate at which we can physically synthesize and verify new biological cures. This shift toward autonomous biological research introduces significant biosecurity risks. Current government regulations rely on humans overseeing humans. Those rules are entirely unprepared for a reality where an AI can independently design a pathogen and instruct a robotic arm to synthesize it before anyone notices. We are caught in a tight tension. Patients desperately need these high-speed cures, but we lack the safety protocols to contain the artificial intelligence designing them. We have reached the point where genetic diseases are solvable engineering problems. The pace of scientific discovery has pulled the future of medicine into the present, and our next challenge is figuring out how to